These are my disclosures. For those of you not familiar with City of Hope, 100 years ago we were a tuberculosis sanitarium and evolved now into one of the great medical centers in America. The biologic response modifiers are listed on this slide, and we're going to go through each of them, starting with monoclonal antibodies, which actually were in the forefront of this area. So back in 1979, when I first started my fellowship and my research project was to make monoclonal antibodies against lung cancer antigens, I put together this cartoon. And it showed that there were targets on the cancer cell that either are restricted to the cancer cell or they're present on normal cells as well, but seen an increased number on the cancer cell, like proliferation-linked determinants. And Drs. Kohler and Milstein in 1975 had developed the technology which led to a Nobel Prize in 1985 to make hybridomas that would secrete antibodies that you could use to target antigens. And in our case, we're interested in making antibodies against human tumors. And so you would immunize a rodent with a human tumor. You'd harvest the spleen cells that saw the antigens on the human tumor as foreign and could make antibodies against them. And you would fuse these spleen cells to a mouse myeloma line that had the capacity to continue to replicate. And it actually had an enzyme defect that allowed you to kill it off in tissue culture. That missing enzyme, when you fused the spleen cells to the myeloma cells to create the hybrid, was inherited from the spleen cells. And if everything went well, you would end up with a hybrid making antibodies, and the spleen cells that didn't fuse would die off. And these antibodies would then be screened to see, did you make an antibody of clinical utility? In my case, against lung cancer. And so this is a cell line. The cell line would replicate every day, and they'd secrete thousands of antibody molecules into the serum every minute. And the antibody molecules are the traditional immunoglobulin molecule with the amine portion, the variable region for antigen binding, and then the FC or constant region for effector functions. In this constant region, you can also link isotopes or chemotherapeutic agents. So how do we use antibodies right now to treat cancer? They're both naked antibodies, meaning nothing's added to them, and there it's immune-based, like directing T-cell killing or NK cell killing or activating complement or interfering with critical growth factors that are necessary for a tumor to grow, or the receptor that growth factor binds to by blocking it. And more recently, and I'll say this in more detail, bispecific antibodies that bind both the cancer and the effector cell, and so they bring the T cell right to the cancer. And then there's all these engineered antibodies where we link poisons, radioactivity, biotoxins, or prodrugs to kill the cancer. So from the time I started my work, you can see this laundry list of antibodies that are now approved, the first being Rituxan, which recognizes the CD20 antigen on B-cell lymphomas, revolutionized the care of patients with non-Hodgkin's lymphoma and some other entities like chronic lymphocytic leukemia. I'm sure you're all aware of the major breakthrough in breast cancer with the antibody against Herceptin, which had a profound effect in controlling the disease of HER2 positive of women as examples. And then you have the conjugated antibodies. It's a smaller group, but this is going to explode in the next few years. The first two were radioactive isotopes linked to antibodies, both against the CD20 target. It turns out when you use radioactivity, you can't use humanized monoclonals. All the antibodies I showed you before have been humanized so they can stay in the circulation and also are less immunogenic. That technology, ironically, had been developed by Art Riggs at City of Hope, and that's what led to this enormous royalty stream. But when you're using radioactivity, you want a very short half-life in the circulation, so these are still the mouse monoclonal antibodies. Two breakthrough drugs that have been just remarkable is brentuximab vedotin. This is against CD30 positive malignancies. You're going to hear more about this by Jasmine Zain with a toxin called auristatin, very effective in this setting. And then in patients who are HER2 positive, you have the adotrastuzumab. Amitriptyline is the toxin used in this instance. Recombinant toxins. So there's only one. It's denileukin diftatox, developed back in 1999, but there's a new version that's now going to soon be available. And here you're linking the protein that binds to the IL-2 receptor, so the IL-2 molecule, to the diphtheria toxin. And the diphtheria toxin has ADP ribosyl transferase activity. And as a result, you inject it into the bloodstream, it finds the cancer that expresses the IL-2 receptor, it gets endocytosed, the active fragment, the diphtheria toxin gets released, and it inhibits protein synthesis, actually by affecting the ability to add proteins through the elongation factor, and then it leads to cell death. And here's one of our cutaneous T-cell lymphoma patients before and after therapy with this product. Vaccines. Vaccines are going to start to see their heyday. Irradiated tumor cells historically have been one approach. I still have a miracle patient on the original trial who had metastatic disease in the liver, and where we would just chop up his cancer, mix it with a adjuvant BCG, immunize him, and he went into a sustained remission and actually retired to San Luis Obispo, which is somewhere around here, I know. Then another approach is to use dendritic cells. So dendritic cells are professional antigen-presenting cells. They take up antigens and they present them on the cell surface. And so you can put tumor lysates or a viral vector with tumor antigens or peptides that have a tumor antigen into these dendritic cells, and then they can stimulate an immune response. There are a variety of adjuvants that can be linked with tumor antigens, like BCG, that'll stimulate an immune response. And then a number of investigators are looking at oral approaches with carriers like lactobacillus, where you have tumor antigens embedded in this bacteria that then get expressed and the body reacts against them. And so there are two vaccine therapies that have been approved. One is BCG for intravesical treatment of bladder cancer. It's considered a vaccine because it stimulates an immune response. And then more recently, Provenge, where you essentially collect blood on 3 Q2-week occasions. This is in patients with prostate cancer. The target antigen is prostate acid phosphatase, and it's actually linked with GM-CSF. And after collection of the samples, the dendritic cells are expanded out of the body. They're infused into the patient and theoretically take resting T cells to be activated T cells. And these activated T cells, though, go and kill the prostate cancer. Adoptive immunotherapy. The simplest version is non-myeloablative stem cell transplant, where you're not trying to treat with intense chemotherapy, you're only trying to immune suppress the patient so that the donor's marrow can engraft. And so if this is your patient who's got leukemia, residual leukemia, and this is the recipient, and then you do your treatment and you do your high-dose therapy and you infuse the donor blood cells, ultimately the donor blood cells start to replace the recipient's blood cells and they start to kill the residual leukemia, and at the end you end up with donor blood cells and no leukemia. This is now one of the most fascinating advances that relate to an allogeneic approach, and this is CAR T-cell therapy, where you take T cells, CD4 and CD8 T cells, and you put into those cells the genes for an immunoglobulin molecule that can recognize a target antigen. And the most compelling results have been with antibodies recognizing CD19 in leukemia and lymphoma. And so these CAR T cells are infused into the patients, and even patients who have far advanced disease and have not entered a remission, and there have been compelling responses, many lasting years. There have been toxicities associated with it, in particular cytokine release syndromes, which are of concern, and also some central nervous system problems. Immunomodulatory drugs. So here you see there's a slew of them. The most well-known are the IMiDs, that's thalidomide, Revlimid or lenalidomide, and then most recently pomalidomide, which are approved for myeloma but also used in CLL and in some of the non-Hodgkin's lymphomas. In addition, you have here the checkpoint inhibitors, that's ipilimumab, and also Keytruda and nivolumab. And in addition, I'll mention BiTE therapy. So for the IMiDs, they have a host of effects in the microenvironment. We don't have time to go through each of the different alterations, but they have effects on every immune cell, and ultimately it's felt that this results in control of the tumor. It's recently been shown, actually through a cell line system we developed, that there's also a target within the cancer cell that they can cause death of the cell, and that's called cereblon, and that is a critical molecule also in the thalidomide-induced phycomyelia that was seen, the heterogenetic effects where those young children were affected as a result of their mother taking the drug during her first trimester. We also have the checkpoint inhibitors. This has been the biggest surprise for me, but it's been a home run. We know that the immune system is trying to fight off the cancer. What wasn't clear is how effective the cancer is in blocking that immune response through signaling pathways. And now we have antibodies that can block these inhibitory signals, and as a result, there's a dramatic response that occurs through T-cell stimulation and death of the tumor in a whole spectrum of solid tumors and blood cancers, including most recently Hodgkin's disease, with over 90% response rate. The main side effect in particular for the CTLA-4 antibody is that you create an autoimmune process within the body, because now these cells are also reacting against some normal tissues in the body, and sometimes it can be quite severe. In fact, we've had several patients who've had bowel perforations from colitis, which is autoimmune. So these are not innocuous drugs, but they're producing very durable responses in historically difficult to treat diseases. And then there's Blinatumomab, and this is like doing CAR T therapy, but not having to do ex vivo manipulations. So what they're doing here is they're injecting an antibody that has two ends. One end recognizes the tumor antigen, and that happens to be CD19, and the other end recognizes the antigen on the T cell, which happens to be CD3, and this is given as a continuous infusion, and you can see these very high response rates and also response durations that occurred in patients receiving these drugs. City of Hope actually did some of the pioneering work here, and many of these patients subsequently went on to transplants. Interferons and cytokines, these are sort of the older versions of biologic therapies. They've been around a long time. Even though we don't use high-dose interleukin-2 that frequently, I have many patients who had disseminated melanoma and renal cell cancer who are alive today 10, 15 years later because of these high-dose therapies, which presumably were stimulating an immune response, though that still has some skeptics. Then we have interferons. Interferons are now given subcutaneously. There's a whole host of diseases where they're effective. They never became as popular as anticipated because the side effect was making the individual just feel crummy, just like you had the flu, and the flu in part is those symptoms are caused in part by the release of interferon. And so interferon is still a reasonable therapy for certain diseases, but with all the modern approaches I'm going to show you, it's sort of moving its way to the back of the line. And that's the interferon molecule. But it was one of the first cytokines we used in the clinic, and we were able to then learn about these signaling pathways and the importance of the signaling pathways in causing growth inhibition and anti-tumor effects. And we learned that if you just eliminated one of these pathways, if there was a mutation or a problem, all of a sudden the interferon wouldn't work. And so it was the importance of having this intact system. Hormonal agents, they've been around a long time. They fall into two versions. There's the version that's aligned with estrogen and androgens. So estrogen stimulates the growth of breast cancer, androgens stimulate the growth of prostate cancer, and there we either shut off the production of the estrogen, like the aromatase inhibitors, or the androgen, or we block it at the receptor level. And so tamoxifen, as an example, blocking estrogen's binding to its receptor. The other version are hormones that proactively kill, like glucocorticoids, which we use to treat a variety of blood cancers, or retinoids, which we use to treat certain blood cancers. They actually bind to specific receptors, enter the cell, and act as transcription factors, turning on and off genes. They also bind to other transcription factors, interfering with the expression of certain genes. And we still have to iron out the exact pathways involved, but they're very effective in inducing cellular death. And so here you see a list of the hormones that we use in the clinic. The mother of all hormone therapies is tamoxifen, and used for breast cancer and anti-estrogen. You see here some of the drugs that have been used in both breast and prostate cancer, either to shut off hormone production or block it at the receptor level. Targretin here is a rexinoid used in cutaneous lymphomas that binds to the retinoic acid receptor, and there's a whole slew of second and third generation hormonal agents that you're going to hear about in the individual presentations. Signal transduction modifiers. This is probably the group that's got the most attention in the last decade. And so the signaling in a cell, any cell, and I'm just showing you a sliver of a cell, is quite complex with many pathways, many redundant pathways, a lot of feedback. But nevertheless, sometimes when you have a mutation or abnormality, which is common in a cancer, and you use an inhibitor, you get a dramatic reduction. And so the next 3 slides literally list all the signal transduction inhibitors that have been approved since 2001. The first being imatinib or Gleevec, which was used for chronic myelogenous leukemia, where there was a specific chromosome translocation associated with a specific gene, BCR-ABL, and if you inhibit that particular gene, you get dramatic killing. It turned out that it also bound to c-KIT, and a rare sarcoma called a GIST tumor, gastrointestinal stromal cell tumor, overexpressed or mutated c-KIT. And I had one of the first patients in the country when I was in Chicago who had one of these tumors, was told nothing could be done. His liver was replaced with disease, and he was the second patient on George Dimitri's trial out of Dana-Farber with the Gleevec compound, but here not targeting BCR-ABL, but its other target, c-KIT, and he's been in a remission for 12 years. So it's quite remarkable. I'm not going to go through the laundry list of all these. They're available to you, and you're also going to hear about these in other presentations. I was just going to comment, though, that there are certain targets that, even though this BRAF inhibitor was developed for melanoma, we get BRAF mutations. It turns out that a blood cancer, hairy cell leukemia, 100% of the patients have a BRAF mutation. And in this issue of the New England Journal of Medicine, there's an article showing the very, very high and durable response rates using BRAF inhibitors in hairy cell leukemia. I'll also mention down here the ALK gene and ALK abnormalities, which is a kinase, was first noted in T-Cell lymphomas. But the first drug that inhibits it was developed in non-small cell lung cancer, where there's mutations in a small population, because it's a larger population of patients. And so as a result, this is how it was developed, but now we've also used it in the blood cancers, where it's a spectacular success. Transcriptional regulators. So here we know quite a bit, but we had a lot to learn about how gene expression is controlled. We do know that genes are methylated in a different way in cancer cells than normal cells, which can often suppress the expression of certain genes. And there are drugs that interfere with this methylation process, 2 examples being acetylene and decitabine, and they're very effective in treating myelodysplastic syndromes and AML. Another example, and this is what we refer to as epigenetics, another example is histone acetylation, which regulates gene expression. And what happens is you have transcription factors binding with the histone acetylators. The histone molecules become acetylated, and they activate chromatin. They let chromatin be expressed. And then if you get deacetylation, it again represses chromatin. So the drugs in the clinic are blocking this process and leaving it in the activated state. And for reasons we don't quite understand, they're very effective in particular against T-cell lymphomas. So the drug vorinostat or romidepsin, or more recently belinostat, all very effective and approved in T-cell lymphomas. Recently, panobinostat was approved in myeloma, but using it alone, the response rates are modest, but using it with similar drugs that are used in myeloma, it seems to enhance response rates. And then there's one of these epigenetic modifiers that's actually quite specific. So we also have histone methyltransferase inhibitors, and they actually work at the lysine group. And the one that's available is produced by Epizyme. It's called tazemetostat, and the target is one of the transcription factors, EZH2. And it turns out the EZH2 is associated with a number of cancers and driving replication. They initially looked at it in tumors that were I1 negative, and these are a variety of sarcomas as an example, because that particular pathway tends to have an adverse and blocking effect on the ZPZ2 pathway. So it's the yin and the yang. And so in these tumors, they've found significant responses. More recently, they've shown overexpression and mutations in non-Hodgkin's lymphoma and success using these agents in that setting. Antisense and microRNA compounds. So none of these are approved, but they have great potential. So double-stranded DNA goes to single-stranded RNA, and if you were to make an antisense oligonucleotide, so this would be essentially DNA bases that can bind to the RNA, and they're complementary to the RNA, you'll get binding, and then there are enzymes that degrade double-stranded RNA, and you stop producing the protein. So one of the targets has been STAT3, a very key signaling molecule, and remissions have been witnessed with this therapy. Another approach is to use non-coding microRNA. So typically, a gene is transcribed to a messenger RNA, and then it's translated into a protein. Well, these non-expressing microRNAs, they will bind to the messenger RNA. They don't code for a message itself, but they'll bind to a piece of RNA, and as a result of this binding, they block the ability of the cell to produce the protein. And so they're actually regulating the expression of proteins in cells. And it turns out that most cancers have signature patterns of microRNA, and that you could use it therapeutically. So we're going to soon open a phase 1 study focused in cutaneous T-cell lymphoma. This is a terrible skin lymphoma. We have a very large population at City of Hope. And MRG-106 is an oligonucleotide that inhibits this specific microRNA that seems important in the development of this disease, the 155. And so we'll be injecting this antagonist to this microRNA, and it's already been shown to reduce proliferation and induce apoptosis. And the genes that are affected in this therapeutic process are STAT3, JARED2, SHIP1, and BOC1, all involved in cell replication, and this trial will soon open. Angiogenesis inhibitors. Judah Folkman, who was a surgeon at Harvard, had postulated that if we blocked the neovascularization that occurs with cancers, we could kill cancers, because cancer needs blood to grow. It creates, in fact, once it's more than 2 millimeters in size, you can't just get the nutrients from the microenvironment. And so there's a neovascularization that occurs as a result of the cancer, and it's been theorized then by blocking this, you'll be able to prevent metastases. And so he had developed 2 compounds which unfortunately never became clinical realities, and it's a tragedy that he's not lived long enough to see everything that's happened in the field that he really was the pioneer, because now we have a whole host of these angiogenesis inhibitors. Some of them are intravenous, some of them are oral. They work against different targets, many against the vascular endothelial growth factor receptor, and in particular in some of the GI cancers, they've had a profound impact. Proteasome inhibitors. So proteasomes are present in all cells, and they're part of this ubiquitin proteasome pathway. They have an essential role in regulating intracellular concentrations of specific proteins. So that means that they control protein homeostasis. And when you want to get rid of a protein, you get this ubiquitin tag that's placed on it, and then it goes into the proteasome and it's degraded. It's like a garbage disposal unit. Well, there are now inhibitors that have been developed against this organelle, the first being bortezomib and the second being colfizimib. And Both are very effective in myeloma. They've been remarkable advances in those diseases. This is a irreversible inhibitor. This is an irreversible inhibitor. The beauty of the carfilzomib, it has less neurotoxicity associated with it. Though this is currently being given IV, I'm sure in the future they'll switch to sub-Q as well. But well tolerated, no hair loss. Neuropathy had been the previous concern. Metabolism inhibitors. So it's been known for 100 years since the work of Otto Warburg that cancer takes up sugar more avidly than normal cells. And that's reflected here in the glycolysis pathway and the Krebs cycle. And so it turns out that if you interfere with glucose uptake, you kill cancer cells. We've taken the tack to block glucose uptake at the surface of the cell by interfering with the GLUT molecules, which essentially suck up the sugar. But others are looking at alterations in the Krebs cycle. It turns out that one mutation that's common in acute leukemia and in brain tumors are mutations in IDH1 and 2. And as a result, rather than producing alpha ketoglutarate, you produce 2-hydroxyglutarate, which ultimately affects methylation patterns and stimulates cancer growth. And so inhibitors to those targets have been shown to be very effective in the clinic. DNA repair inhibitors. We have one that's approved, olaparib. It's oral. It's for women who have ovarian cancer and who also have a BRC mutation. And its mechanism of action, it's a PARP inhibitor. So if you normally damage DNA, and you can do that through oxygen-free radicals, that can be, say, through chemotherapy, spontaneous reactions, X-rays, like the bombing of Hiroshima, or alkylating agents, you get single-strand breaks, and they're repaired in a normal cell by either base excision repair, that's the BER, or by homologous recombination repair, the HR. And as a result, you get DNA repair in a viable cell. You can be missing the homologous repair in someone who carries a BRCA mutation, but still the cell is viable because you have the base excisional repair. If you inhibit PARP, then you inhibit the base excisional repair pathway. And so for patients who have breast cancer or ovarian cancer, and there's a report in the New England Journal of Medicine this week on prostate cancer, who have this defect, if you then inhibit the base excision repair pathway using a PARP inhibitor, you can't repair the DNA and you kill the cell. And so it's a new avenue of therapy that's being developed. Apoptosis inducers. So again, the cell signaling is quite complex, but what we do know that those molecules in the box, the BCL2 molecules, their role is to prevent cells from dying. And they're very effective at doing that. And so inhibitors have been developed to BCL2, and at least in chronic lymphocytic leukemia, the remissions have been simply dramatic. RNA-based analogs, and this is near and dear to my heart. This is the work I do, and we just received $4 million in grants to support this work. So I'll use the 8-chloroadenosine as an example. So this looks like an adenosine molecule, like the molecule that becomes ATP, but it's got a modification here. And so it enters the cell. It could be deaminated, but hopefully in the cancer cell, it goes through a variety of kinase steps, ultimately becoming 8-chloro-ATP. So again, looking like ATP with this modification. And it incorporates into RNA, and at the same time, it depletes normal ATP levels. So here you see the decline of normal ATP levels. You see the rise in 8-chloro-ATP in the cell. And it has very little effect on DNA in contrast to all the other nucleoside analogs that are used in the clinic, but has a profound effect on shutting off RNA production. And so cells don't have to be replicating in order for it to work. And most of the cancer cells aren't replicating very fast. And so we're very excited because in vitro, we've been able to show it has profound effect against patients with acute leukemia and actually some other tumors, and even against those that have mutations that have been historically challenging to treat versus normal cells. Oncolytic viruses. This is the hot topic of the moment. This is the most recently approved agent. I can't even pronounce it, but it's a herpes simplex virus that has GM-CSF linked to it. Randomized trial in melanoma showed profound benefits. These viruses enter the cancer cell and the normal cell, but they don't replicate in the normal cell, but in the cancer cell, they replicate and lyse the cancer cell. There's been reports of poliovirus in brain tumors as an example, and then there was this remarkable report of a measles virus creating a sustained remission in a patient with myeloma, where the measles virus is manipulated in a way it can't replicate in normal tissue, but only in the cancer. Three quick ones. Bromodomains, these are the newest kids on the block. You can see all the diseases they're effective against: leukemia, neuroblastoma, glioblastoma. Bromodomains are proteins that bind to specific acetylated groups on transcription factors, and they'll bring in other key molecules in order for genes to be transcribed. How they're actually working still remains a little bit of a puzzle, but they are working. And then telomerase inhibitors. Telomerase inhibitors, and the telomere is at the end of the chromosome. It's like the little plastic thing at the end of your shoelace. It sort of protects it. So these telomeres protect the end of chromosomes, and they're repetitive units. And if you didn't have them, the chromosome degrades or fuses with another chromosome. Turns out in cancer cells, these telomeres, though shorter, there's more activity of an enzyme telomerase, which keeps them going. And so if you inhibit telomerase, you end up killing the cancer cell, and you don't affect the normal cell. And there were two reports just recently in the New England Journal, one with the treatment of myelofibrosis. This is the compound Methylstat, and also in essential thrombocytopenia, showing the benefits of these inhibitors. David Snyder at City of Hope was one of the lead authors on this report. And last, nuclear export inhibitors. There is a compound being used, Selexant 4. Turns out that there are pores in the nucleus. There are proteins that are shipping out proteins in the nucleus constantly. For reasons that aren't clear, a lot of the proteins that are being shipped out are the tumor suppressor proteins, and so inhibitors to this molecule, XPO1, which Selexant is, they interfere with this process, and you get an accumulation in the nucleus of these inhibitors like P53. And so these have been shown to be effective in melanoma, prostate cancer, non-small cell lung cancer. In addition to everything I showed you in therapeutics, there's an equal array of phenomenal compounds in supportive care that have benefited our patients. And coming soon, because of molecular profiling, God knows how many additional agents we're going to have that may benefit our patients. And with that, I'll say thank you for your attention and apologize for running over.